Lesson 2. Sample integrity. If you are requested to collect a urine sample to diagnose if you are suffering from UTI, what kind of container you will use? How do you collect it? You firstly pour it in a container and then pour it to another container, or you directly pour into it. How much will you collect? Just a half bottle, a full bottle, or just a little bit? What should you do after your collection? So how do you store it? Just put it on the table in room temperature, or put it into fridge, or just directly take to the laboratory once after you collect the sample. So actually, sample integrity is a very important issue in clinical laboratory. Test result is meaningless if it comes from a poor quality sample. In the pathology lab, the general workflow can be divided into 3 parts: pre-analytical, analytical, and post-analytical. In the pre-analytical, it involves of test ordering, sample collection, the transportation of the sample, and it reached to the lab. We will do the lab test registration, and then we will prepare the sample for analytical part. Before we doing the test, we will firstly have some maintenance and daily check of the analyzer, and then we will do calibration if it is needed. We must do quality control to ensure the analyzer can be functional and can provide accurate results. So if the QC quality control is passed, if the quality control pass, we will test the sample. If quality control fail, we will troubleshoot, recalibrate it, and then do the quality control again until it is passed. Then, until the quality control pass, then we will test the sample. After testing, we will post to the post-analytical part, which we will verify the results to ensure the result is valid. Not just for the quality, but if it's matched to the patient's previous history, or the whole profile is it matched. So once the result is verified, then we will report it to the ward or to the clinician. So in the whole process before we do the analysis, sample collection is a very critical part. We have to ensure the sample is good in condition. We have to ensure the sample collected is in. We have to ensure sample integrity before we process. We have to ensure sample integrity before we proceed to analysis. Actually, all the biological materials can be collected to be tested. Actually, all the biological materials come from your body can be tested. But the most common biological sample, but the most common clinical sample is blood. Other than that, body tissue, urine, feces, body fluids, and sputum are also common samples. Are the other common samples. Other than blood, body tissue, urine, feces, body fluids, and sputum are the other common samples. So in the previous lesson, we introduced 4 main disciplines in the pathology lab, which is hematology and blood bank, chemical pathology, medical microbiology, and anatomical pathology. So in these laboratory, there are some common types of sample. For example, in blood bank, we use is whole blood and bone marrow aspirates. In clinical chemistry, we also test blood, but we will centrifuge it and collect. So in the previous lesson, we introduced in the pathology lab there are 4 main disciplines. Each discipline have its common types of specimen, but you can see blood is the most common type of sample. In clinical chemistry, in the chemical pathology lab, the whole blood sample will be centrifuged to yield so blood sample is the most common type of sample. The other one is urine. Sputum is also common. But in chemical pathology, the whole blood sample or the blood in chemical pathology, the blood sample will be centrifuged and yield with plasma and serum. So in chemical pathology, we will centrifuge the blood and yield the plasma or serum for analysis. And the urine, in chemical pathology, we usually is random urine or 24-hour urine. In microbiology, and for the urine sample, it is not just collect randomly or collect all of the sample. In chemical pathology, we usually collect random urine or collect 24-hour urine for analysis. But in medical microbiology, we will collect the midstream urine or the early morning urine, which is concentrated. We will use midstream urine, which is only the middle part of the urine without any contamination, or EMU, early morning urine, which is much concentrated. And about the blood, we commonly received is capillary blood or venous blood. We usually collect capillary blood from heel or finger puncture. We will collect capillary blood from heel or through finger puncture. The first drop of the blood should be discarded. So the first drop of blood should be discarded. So the blood won't be mixed with the lymphatic fluid. And they are collected in the micro containers, so a small amount of blood is about blood. We commonly collect capillary blood from about blood sample. We commonly collect capillary blood or venous blood. Capillary blood usually We usually collect capillary blood from children or neonates. So the blood is collected by heel or finger puncture. The first drop of blood should be discarded in order not to mix with lymphatic fluid, so the blood won't be contaminated by lymphatic fluid. So the blood won't be diluted and contaminated by lymphatic fluid. And they are collected into these micro containers with a small volume. About venous blood, we should collect in appropriate sites to avoid any contamination or dilution by infusion. So we never collect blood from drip arm. And then when collecting the blood, we should follow a correct blood drawing techniques and order. And also we should follow the correct blood drawing techniques to collect the blood in correct blood tubes and in correct sequence. The sample should be delivered to the laboratory less than 2 hours from collection in room temperature, if it is stored in room temperature. If it is not, the sample should be delivered to labs. The sample should be delivered to the laboratory within 2 hours from collection. If it is not delivered once after collection, samples should be delivered to laboratory within 2 hours from collection, or it should be stored properly. For example, put it into refrigerator if necessary, according to the guideline. And this is the correct sequence of blood drawing. The sequence of because each of the blood tube inside there are some additives, for example anticoagulants or some activator to activate the clotting process. So we should follow the order in order to prevent any contamination or carryover of the additives. If contamination occurs, that will affect the final results. If there is any contamination or carryover of the additives, there will be false results. There will be false results. So actually, the whole blood can be centrifuged in order to yield two parts. The superlatant can be serum, if it is a clotted blood, or can be a plasma if it is a whole blood. If you can see the supernatant becomes red in color, it is a hemolytic blood. It is a hemolytic sample. Because the hemoglobin inside the red cells is lyed, because the red cell is lysed and the hemoglobin is released. If you can see the supernatant is milky in color, it is due to the presence of fats. It is a lipemic blood. Sometimes you can see there is brownish yellow color of the supernatant. It is due to the increased level of bilirubin. We say that is etheric blood. So all these three types of blood sample can affect some of the tests, which depends on the all these three types of sample can interfere some of the tests, which depends on the calorimetric method. All these three types of sample can interfere the results All these three samples can interfere some of the test results, which depends on colorimetric method. All these three kinds of sample may interfere some of the test results, which depends on colorimetric methods. Urine sample can be There are roughly four types of urine sample. Random urine is the most common type of sample. It can be collected at any time. 24-hour urine is collected within a 24-hour period. Let's say, for example, the first fording at 8. Let's say, for example, the first morning urine at 8 p.m. For example, patient is instructed based on instruction, the first morning urine will be discarded. For example, at 8 a.m. Then all the other 40 urine will be collected into the big container until the next morning, 8 a.m. Collect the final one. So all 24-hour urine will be collected. 24-hour urine meaning to collect 24-hour urine is collected within 24-hour period. All the urine passed within this period will be collected and put into a collection cell to a large container, usually with preservative. Early morning urine is the first urine voided in the morning, so it is more concentrated. For example, for TB, tuberculosis detection, early morning urine will be requested. Midstream urine is commonly used for microbiology culture. Midstream urine is commonly used for urinary tract infection. Midstream urine is commonly used to detect microorganisms in UTI. Cleaned The patient should clean the urethral area before sample collection to avoid any contamination from the skin. To avoid any contamination of the skin flora. And the patient should void and discard the first portion of urine stream and then collect the midstream urine into a sterile container. So when we ask the patient to collect sample, we should provide clear collection instructions and then provide a correct container for them in order to get a proper sample. For respiratory sample, we commonly collect the upper respiratory sample and the lower respiratory sample. For respiratory sample, there are two main types, which is come from the upper respiratory and the lower respiratory. For respiratory sample, it it can be divided. It is divided into two types. One is from the upper respiratory tract, and the other from the lower respiratory tract. Throat swab, nasal aspirates, and throat swab, nasal aspirate, or wash. In the upper respiratory specimen, and for respiratory sample, there are two main types. One is come from the upper respiratory tract, and the other one from lower respiratory tract. To collect sputum, it is more easy to collect it in the morning. To collect sputum, it is more easy to collect it in the morning, and it should be collected through a deep coughing and avoid any saliva collection. When the laboratory receives the sputum sample, we will differentiate whether it is a saliva or a true sputum. We can through macroscopic feature. If it is a saliva, it is watery. Under microscope, we can see that there are many squamous epithelial cells and the absence of any white blood cells. But if it is true sputum, it should be mucoid, fluent, or blood-stained. Under microscope, we can see many WBCs. So to minimize sample contamination or degradation, sample integrity should be maintained through collect the sample in a proper way, and you should preserve the sample in the correct container, and we should preserve sample in the correct container and store the sample in a suitable environmental condition before it is transported to the laboratory. And when there is a loss of sample integrity, for example leakage, inadequate volume, incorrect container, etc. And when there is loss of sample integrity, for example there are leakage, inadequate of volume, incorrect container, or improper storage condition, etc., the laboratory will reject the sample and we will request a subsampling in order to get a proper sample for analysis.